step, we quantitatively assessed the apoptosis using annexin 5 FITC propidium iodide dual staining assay. This kit is based on FITC conjugated annexin, which binds phosphatidylserine in the cell membrane of apoptotic cell, and propidium iodide is the marker of cell apoptosis and cell membrane permeability. So using this kit, we can measure the percentage of viable early apoptotic, late apoptotic, and necrotic cells. And in our experiment, cells were treated with LDN in 2 concentrations. The positive control were cells treated with staurosporine, which is the inductor of apoptosis. Negative control, untreated cells, and additional control, cells treated with DMSO as a solvent for LDN. And we measured the outcomes by flow cytometry. And our results are that cells has no significant increase of percentage of necrotic cells after treatment with LDN after incubation for 3 and 24 hours with LDN at concentration 6 and 50 micromole, and together. Okay, one moment. Yes, the flow cytometry revealed no significant induction of apoptosis by LDN in any used concentration and incubation time. Our conclusions are that small molecule inhibitor of PERK branch of UPR signaling pathway, LDN, first of all, has no cytotoxic effect on DNT and C1 cells, does not induce apoptosis, and might provide neuroprotection in rat astrocytes by inhibiting PERK. So we may assume that this molecular target is a really novel treatment strategy against Parkinson's disease. Thank you for your attention.
Thank you for your presentation, and maybe someone from our jury want to ask some question for you. Okay.
Yeah, hi, thank you for the nice presentation. I just have one question. According to this inhibitor, did you check if this inhibitor really inhibit the pathway?
Yes, we've checked it by the Western blot experiment when we measured the inhibitant activity of PIF2 alpha. So we can conclude this the PERK is the specific molecular target of LDN.
And more or less, how strong was the inhibition? To which level? Can you say it in the percentage? How strong was the? Yeah, because it depends. If you just block the signaling pathway till 50%, then it's still there. Yeah. If you block till 10%, then we can say that it's working. That's why I'm just asking if you check it, or maybe you can check it in the future.
I should check it in my previous presentation.
Good morning. Good morning. My name is Joanna Strzelczyk. I would like to ask about your own contribution to this work.
Okay, I participated in the laboratory part of the research. So I conducted the experiments in the laboratory together with my main tutor, Natalia Siwecka.
Okay, I want to ask for the time points because you choose 3 hours and 24 hours for the apoptosis. Why this time, 3 and 24? Because this is completely, no 6, 12, 24, etc. But 3 and 24, why?
It was based, first of all, on literature in the comparison to commercially available GSK inhibitor of PERK. And of course, we checked after 6 and 12 hours, but the results were not so significant. So that's why we chosen those two in these results in today's conversation.
Someone want to ask any other question? Okay, so thank you very much for your presentation and discuss. And now let's start second presentation presented by Magdalena Król.
Dear ladies and gentlemen, my name is Magdalena Król, and I am a student of Jagiellonian University Medical College. I would like to draw your attention to the research conducted at the Department of Pathophysiology that focused on the cardiovascular implication of long-term fluoropyrimidine administration in a rat model. At the beginning, I would like to tell you about 5-fluorouracil. This is a medication used to treat various malignancies such as head and neck, gastric, and colon cancer. This is also the second most common chemotherapeutic agent that causes cardiotoxicity, and the mechanism of this phenomenon is multifactorial and clinically leads to arrhythmias, vasospasm, and direct myocardial injury with a heart failure as a result. Concerning the purpose of our investigation, we wish to examine the cardiovascular effects of long-term fluoropyrimidine administration in an animal model, and to assess these effects, we used magnetic resonance imaging, and additionally, we analyzed the blood serum samples. The study included 18 male Wistar rats randomly divided into two groups. The first one, the research group, was administered with 5-fluorouracil at a dose of 150 milligrams per kilogram intraperitoneous every 2 weeks. And the second one, the control group, received saline, also intraperitoneous every 2 weeks. In total, every rat got 4 doses of the drug or saline, and the magnetic resonance imaging measurements were conducted before the administration and 3 days after every dose, and the blood tissue were collected also before the administration and after 4 days after every dose of 5-fluorouracil or saline. The good quality of obtained MRI records allowed us for a precise analysis of crucial parameters, and this is an example of that image. And now I will present an overview of the results. On this chart, we may see that the mean ejection fraction decreased gradually after subsequent doses of 5-fluorouracil. Furthermore, the mean peak ejection rate also gradually decreased after subsequent doses. However, the mean peak filling rate was not affected by this, but the concentration of troponin I in the blood samples was significantly increased after the doses. To conclude, 5-fluorouracil based chemotherapy results in morphological and functional changes in the cardiac muscle. This chemotherapy, as our results presented, has a negative impact on this muscle. And in our study, it manifested as a progressive dysfunction in the myocardium, as indicated by decline in the mean ejection rate and the mean peak ejection rate. And moreover, the increase of troponin I concentration also proves the cardiotoxic effect of 5-fluorouracil. And these are the references I used during preparation of the presentation. And thank you very much for your attention.
Thank you very much for your presentation. And now we have 3 minutes to discussion. Maybe someone wants to ask. Do you have any future plans with the next experiments?
Yes, the results I presented are a little part of a bigger project grant we currently do, and we analyze different aspects. Also, we try to find a substance which could be used in the future to adverse to minimize the cardiotoxic effect, and we try to find the molecular base and the mechanism which could be used to find the best substance or even the drug which is used, for example, in the treatment of heart failure.
Okay. Someone else? Maybe some from today's participants. You also can ask question.
Yes, I have the same question regarding the contribution to the study.
Yes, so 3 years ago, I had a special course to get the right to work with laboratory animals. So me and my team, we administer the drugs. We also analyze the blood samples. Only the magnetic resonance imaging was done in the cooperation with Polish Academy of Science. And generally, of course, we also, with my team, we analyze the results and find the conclusions.
I'm curious, what dictated the choice of the particular active pharmaceutical ingredient? And the second one, what dictated the choice of the used animal model? Thank you.
Could you repeat the first question?
Why did you choose the particular active pharmaceutical ingredients? The drug.
5-fluorouracil.
The drug, yes. Why?
Okay. First of all, it was connected with the fact that 5-fluorouracil is widely used in treating various malignancies, but its cardiotoxic effects often limits the possibilities of the treatment just because the patients have adverse effects and cannot continue the treatment, which is very effective, but not always is possible to use it. So it's just for the future in order to find something to help with having these adverse reactions, and in order to find the substance which could be used during the treatment, just the patient would got not only the chemotherapeutic agent but also why rats? Yes, animal model because this. The organism of rat is one of the most animal which is similar to human's organism, and there is well, it why I have to think a little. For sure, our scientific group is at Department of Pathophysiology, so we all focus on the animal model, but also thank you very much.
Thank you very much. Congratulations for your presentation. Thank you for your questions. And now we have a third participant, but online form from Zoom platform. Do you hear us?
Yes. Hello, everyone.
Hello. Just wait a second. We need to check if everything is okay. Okay, you can start.
Okay, thank you very much. Good afternoon. Today I have the honor of presenting some information about Fabry disease, monitoring using the fiber evanescent wave spectroscopy. Could you change the slides, please? Thank you very much. Fabry disease is a lysosomal storage disease. It's a hereditary disease coupled with X chromosome. The gene responsible for coding of the enzyme called alpha-galactosidase has incorrect structure. Hence, the deficiency of that enzyme in the human body would lead to the accumulation of large quantities of single lipids in lysosomes of various cells, such as cardiac or renal. This next one. The main symptoms are disruption in the thermoregulation system, enlargement of the heart and heart failure, and also proteinuria and renal failure. Please, next one. Total urinary protein and albumin excretion has been considered one of the most important indicators of Fabry nephropathy. While diagnosing Fabry disease, our main focus should be in looking for the presence of characteristic genetic mutations, high concentration of lyso-Gb3 in plasma or in urine, and also the activity of the enzyme alpha-galactosidase. And the study presents the result, firstly, I would like to tell that lyso-Gb3 can be detected using physical methods such as tandem mass spectroscopy. And here you can see the chemical structure of lyso-Gb3. It is given below. And please, next one. The study presents the results of the study of human urine samples from 5 patients affected by Fabry disease and 12 healthy donors. A table on the page contains clinical information about experimental patients. Sorry, can you see me right now? Oh. Okay. Sorry, I didn't message. Okay, so let's consider what fuse is. When light undergoes total internal reflection while passing through a fiber at the reflection points on the surface of the fiber, and evanescent field occurs. And evanescent waves interact with the environment and return to fiber. And as a result, using the MCT detector and the spectrometer, it is possible to read information from the environment surrounding the fiber in the ER spectrum. The scheme of such an installation is shown here, and the fiber is in contact with the experimental sample. But we need to remember that human urine consists of about 91 to 96% of water, so it's necessary to dry it. The technology is absolutely similar to the already well-known ATR, but in case of fuse, we can release a much larger number of reflections and accordingly increase the sensitivity. German telluride fiber, deep fiber pieces play the role of a sensor. And can you give me next one, please? We present the results of studies of human urine samples investigated with fuse in the fingerprint spectral region. The following slide shows a table with a decoding of peak position according to the vibrational motion sources substances, and the main interest here is two peaks highlighted in red dashed lines, as well as like in figure 6, which are corresponded to CO and CC symmetrical stretching vibrations related with galactose chains are located generally in the spectral region of 1,000, 1,150 inverse centimeters, as well as other urine components. Therefore, it is necessary to perform complex statistical methods for qualitative analysis. And here we have an illustration of analysis of spectrum at our range. Thus, we can see in figure 7 the average second derivatives of control and Fabry disease groups, as well as statistical analysis. In this slide, we demonstrate the multivariate analysis using PCA methods, and figure 8 considers the 3D score plot with confidence ellipsoids, which show the separation of two groups of samples. Figure 9 represents the result in the probability scale, and it is strictly correlated with PCA plots. All samples of the Fabry disease group are clearly separated from the control group and lie within the high probability of the presence of the disease. And the probability plot was built with two algorithms, LOR and SVM. So finally, on this slide, we have our machine learning calculation results. As we can see in table 3.
I'm sorry, Nicola, I need to pause you because your time is up. So please move to the conclusion.
Okay, so we can stay before, actually. And I would like to tell that, as we can saw in some table, chances of missing a person with Fabry disease are exactly 0.0, and chances of false positive error are also really low. It's about 0.4%, which means that there will not be many extra cost demands. And it's really important for us because, just like I said before, Fabry disease is a really rare disease, and we need to have good biomarkers to find a lot of patients with that problem and illness. So, thank you very much.
Thank you very much, Nicola, for your presentation. And now start the discussion. Maybe someone to ask.
Yes, please describe the clinical application of the study.
Okay, so actually, I'm the medical student from Rzeszów, and my tutor work at internal medicine department in the hospital, and actually, by the accident, she found out that one patient, really old, when we are talking about rare disease, has a lot of problems with kidneys, with the heart failure, a lot of strokes in the young age. And it found out that he is ill and he's got a mutation just connected to Fabry disease. Later, we started to a little investigation with his family, and it turns out that actually also his sister and his aunt and mother just got the same mutation. So we decided to start take care of patients with Fabry disease and other rare disease like Gaucher. But it's still really low number of patients. It's really hard to find a lot of patients with Fabry disease in all of Poland. Approximately more than 20 or 13, it depends on sources. But we are still looking for them because it's still interdisciplinary problem, and a lot of patients have a long journey to be diagnosed well because they are visiting a lot of specialists with different symptoms, but sometimes it's just that illness. I'm sorry, but I can't hear you. Excuse me. Could you turn on the microphone?
Sorry.
Yes, yes. Please, sorry. Please describe your own contribution. And I have the same question or request for all participants about your own contribution.
Okay, so we've been working on our project for about 1 year, and mostly I was taking care of patients, and I was doing physical examination with my tutor. Moreover, I coordinate the exact way for transporting the samples with the urine to our laboratory, university laboratory. And also, I was preparing a control group for our studies.
Thank you very much.
Thank you very much for your attention.
Could you describe shortly, briefly, the criteria of exclusion and inclusion criteria into the study?
You need to turn on your microphone.
Sorry. Actually, we just decided to take every patient who could possibly give us the urine, because, for example, one patient, firstly diagnosed with Fabry disease, got so big nephropathy that he couldn't even give us the samples. So it wasn't any special criteria, just 2 families with Fabry disease from our department, in every age.
Nicola, I'm sorry, we need to stop, but thank you very much for your presentation. Congratulations. And next presentation also will be online in a Zoom platform. Alicja Kamińska will present her presentation. Do you hear us?
Yes, I can see you and hear you, but I am not Radosław Dudczak, so maybe I will share my screen if possible.
Sorry, Alicja, we don't have your presentation.
Okay, so would it be possible to share my screen?
Yes, please.
Okay. So probably you have to stop sharing your screen so that I am able to share my own. Oh, perfect. So now you can see if that's possible.
Yes, perfect. We can see.
Okay.
You can start.
Maybe I will just turn on my camera as well, so you can also see me. Okay, dear committee, dear guests, my name is Alicja Kamińska, and I am a 6th-year medical student at Poznań University of Medical Sciences. And today I would like to present you my research project, which is analysis of drug release from spheres whose degradation depends on metalloproteinases in a 3D model of neoplastic invasion, which I did in the biotechnology department at Poznań University of Medical Sciences. And as a short introduction of my presentation, I would like to tell a few words about metalloproteinases, what they are, So metalloproteinases are a group of proteolytic enzymes that affect the processes of remodeling of extracellular matrix components and play an important role in the development of tumors, forming metastases. And an increased expression of individual MMPs, this is the way I will call metalloproteinases to shorten it, in tumors is widely seen, which is the basis of linking them with clinical prognosis, actually worse clinical prognosis. The second area of my interest is spider silk, and the spider silk is a material with a wide potential application in biomedicine. The use of genetic engineering techniques enable the functionalization of the silk, as you can see here on the left figure, by its chemical modification and introduction of specific sequence domains. And in our project, we use silk domains which are recognized and cleaved by metalloproteinases, which overexpression, as I previously mentioned, is observed in vast type of cancers. This can be used at an advantage in our case, as degradation of silk nanospheres allows the release of drugs into tumor microenvironment. And so far, as you can see on the right side of this figure, in the literature, many chemotherapeutic agents were used to incorporate in spheres like paclitaxel, doxorubicin, methotrexate, and curcumin. So now I will move to my results section. This is the order that I will present my results. So first I will talk about spheres formation, then I will turn to induction of invasion, inhibition of invasion, spheres formation, incorporation of spheres into spheroids. Please do not confuse these two words. Spheres are silk-based and spheroids are cells-based. And then I will conclude my work. We used a live cell analysis instrument manufacturing by Sartorius for our analysis. And the first slide, we first started from the formation of spheroids using two cell lines. On the left, you can see the MDA-MB231 cell line, which is a triple negative breast cancer cell line. And on the right side, you can see the MCF7 cell line, which is the ER-positive breast cancer cell. So we here produced a model of like a 3D model of tumor microenvironment and tumor itself, actually. Here on the second slide, you can see the experiment with induction of invasion in our 3D model of tumor microenvironment on these two cell lines, MDA-MB231 and MCF7. The invasion of tumor cells from the spheroid into the matrix was stimulated by chemokine CXCL12 in this case, and the bottom pictures show the inhibition of invasion using inhibitor. In our case, we use Oridon in this presentation. We optimized the method to achieve the best model of invasion and the best model of inhibition of invasion, testing various concentration of every agent and their combination of one and more. And next, you can see the next part of our experiment is spider silk-based spheres formation. So those are spheres, not spheroids. Those are drug nanocarrier system, which are based on bioengineered spider silk proteins cleaved by metalloproteinases. They were produced in our lab in the bacterial expression system by salt precipitation method, and we confirmed the morphology and size of the nanoparticles using scanning electron microscopy. Those are the same pictures, but this is a little bit on the right side, this zoomed in. Next, we move to spheres incorporation into spheroids. So you can see those red spots are spheres, which are fluorescently labeled on the two cell lines, on the left MDA-MB231 and on the right the MCF7 cells. And here we loaded spheres with doxorubicin, our first trial with the first chemotherapeutic agent. On the left you can see the MCF7 spheroid with the spheres which include doxorubicin in the concentration of 1 microgram per ml. And on the right side, control sphere with control spheroids with spheres, but without doxorubicin. And as a conclusion, we optimized the protocol for formation of a 3D spheroids model based on 2 cancer cell lines to mimic the tumor microenvironment. And thanks to metalloproteinases, we could support the cancer invasion and see how it is going. The second conclusion is that we optimize the protocol for efficient incorporation of silk spheres into spheroids models. And in the future, we plan to study the controlled drug release more than doxorubicin, but also other chemotherapeutics agents, and also start in vivo studies. And the third conclusion is that we also design a nanosystem formed by biodegradable spheres, recognizable and cut by metalloproteinases, in our case, specifically 2 and 9. This is my contribution. I was responsible for planning and conducting the following experiments: spheroids formation protocol, spheroids invasion and inhibition of invasion. I was responsible for analyzing and do all of the microscope images. I was responsible also for planning and conducting the spheres incorporation into spheroids. And also I achieved the grant support for my work from my university. And I would like to thank you, my all tutors and support from my department. Those are my references, and thank you very much.
Thank you very much, Alicja. Let's start the discussion. Someone want to ask? Okay, we have a question from another participant.
Yes, hello. Did you try to specify the exact metalloproteinase isoforms expressed in your model?
Yes, we did such an experiment in those 2 cell lines. I didn't include them here, but we observed that the MDA-MB231 cell line has much more metalloproteinases than MCF7 cell line. So I don't know why I couldn't resume my presentation. Maybe I am not technically the best, but if I could show you my first slides with the spheroid formation and invasion, you could see that the invasion on MDA-MB231 cell line is much bigger. Like those lines coming out from the spheroids are longer in MDA-MB231 cells, which means that the cell line is characterized by more metalloproteinases 2 and 9.
Okay, thank you. Someone want to ask a question, maybe from online jury? Okay, no one wants to ask a question for you. So thank you very much for your presentation. Congratulations.
Thank you.
And now, please start the 5 presentation presented by Maria Turkiewicz, Szymon Nowak, Lidia Ziobro, and Szymon Stanula. Oh, but we don't have your presentation. Okay, good.
No.
Good afternoon. My name is Maria Turkiewicz, and here with my colleagues, I would like to present to you our presentation about the effect of spions on the cancer cells captured in the human body. We are representing the Jagiellonian University Department of Chemistry, Department of Biochemistry, Medical Biochemistry, and please. Previous slide, please. Okay. So we are, our experiment included breast cancer cells, and as we know, cancer is quite a big challenge currently in modern medicine, and we picked breast cancer cells because it is one of the leading causes of cancer deaths in our population. And also because of the clinical applications of our spions, which I will explain in a moment. Okay. So spions are super paramagnetic iron oxide nanoparticles. So these are iron oxide particles that are coated with several polymers, or in our case, it was chitosan, and this coating can improve some properties such as impact hydrophilicity or impact the way they are captured by macrophages in the liver. And what is important here is that spions show magnetic properties only in the presence of a magnetic field, so they are super paramagnetic, which can actually give us some quite interesting potential applications. They were obtained by Jagiellon University Department of Chemistry students. So they conducted the synthesis. It was the co-precipitation of iron oxide with chitosan chloride. Thank you. And then they examined spion properties in order to determine whether they are appropriate for further research. So they examined their size, they examined whether they are in fact coated with chitosan. As you can see, they determined that the average size was about 230 nanometers. And they analyzed the surface and they analyzed the magnetic properties. And potential applications of spions are actually quite interesting because of their magnetic properties. They can be used as MRI contrast or inducing hyperthermia locally after being injected intravenously into the human body and then applicating an alternate magnetic field. Our aim was to couple them with antibodies potentially in the future. And this is, our presentation is the previous part of that research. So after being coated with antibodies, they could potentially be used to guide, being guided magnetically and capture cancer cells circulating in the human body, preventing metastasis. So this was our aim, finding a way to reduce the general toxicity of cancer treatment by doing it a more targeted therapy. We used some biological testing methods to first determine the spion properties, because they are supposed to be used as carriers, but in order to use them as carriers, we first need to examine how they themselves impact cancer cells. So we assess their cytotoxicity, their impact on cell proliferation, and their impact on some protein expression, Here you can see microscopic images of spions after being incubated for 24 hours. This is the T47D cell line. And here is some pictures of spions after being incubated for 48 hours. You can clearly see the conglomerates of spions around the cells. And when it comes to cytotoxicity, we used two assays. We used MTT assay and lactate dehydrogenase assay to determine whether they show cytotoxic properties, because we do not want them to be cytotoxic, so we need to check that first. And as you can see, the difference between different concentrations of spions and the control group were pretty minor. In higher concentrations, we can observe some bigger, for example, percentage of dead cells in the lactate dehydrogenase assay. However, this is important information, but these are quite minor changes. And then we assessed cell proliferation using the crystal violet assay. So it shows the change in cell adherence, because obviously cells that do not proliferate are not adhered to the wells in the experiment. So the crystal violet assay showed some differences, as you can see, between the control group and the spion concentrations. However, if you take a look at the standard deviations marked on the chart, the differences are not quite as big, but we need to observe that in higher concentrations, such as 10 micrograms per milliliter, it was higher. And finally, we ran a Western blot trial to check for some protein expression. So we ran the test for these proteins listed here. As you can see, they do slightly impact, for example, the expression of BAX in higher concentrations. What is concerning is the low expression of N-caterin, because it would be our potential target for antibody capturing. However, right now there is a third cell line will probably be included in the research. That has a higher expression of N-caterin, so we will also examine its impact on that. So to conclude, our experiment revealed that spions do not show major cytotoxic properties. They do not massively impact cell proliferation. They impact it minorly. And they do not significantly change the protein expression. Therefore, we can conclude that the research indicates they could be used as carriers for antibodies or in maybe other research drug carriers. But our aim is to coat them with antibodies. So we are planning to continue our research furtherly in that direction. Thank you.
Okay, congratulations. Thank you for your presentation. And now somebody maybe want to ask.
Yes, I have a question regarding results in other works. Could you shortly tell us about this? Other publications and the results.
I'm sorry, could you please explain what you mean?
In your work, you check the spion in the breast cancer cells. In other works, in other publications, what was the goal, the aim of the study, and the results?
I truly apologize. I don't think I understand the question. I can say that at our department currently, it is a part of a bigger study in which prostate cancer cells or pancreatic cancer cells, as well as melanoma cells, are also examined, and our part is breast cancer cells.
Okay, thank you very much. And about your contribution?
So we conducted the laboratory work mentioned on the presentation.
Yeah, and I additionally prepared some of the charts with data and analyzed some of those. So yeah, that's it also.
Oh, thank you. What is the characteristic of those cells? Because then I see you are talking about EMT phenotype. MCF7 for sure is not the invasive cell line, right? And what about the second, this
T47D? They are, and the characteristic property of them is that they are not sensitive to progesterone. So that is definitely, but I am not sure how to answer the.
And are you planning also to use some cell lines which exhibit the EMT phenotype, like MB231? Or?
Yes, we are planning to use them. Currently, we already started the experiment.
Because it would be nice to compare MCF7 and MD.
Yes, it is currently.
Well, also the aim of assessing the protein expression was sort of to check whether spions can facilitate EMT. So also that's why we chose cell lines that normally do not show this epithelial imaging hemotransition.
Yeah, but for the control, it's nice to have cell lines which is completely opposite.
Of course,
yeah, to prove that your
yes,
hypothesis is right.
We started this research actually right now.
I want to ask for the negative control because everywhere here you show the effects, but I don't see anywhere here any negative control. Is there were anything, something like this?
Yes, in fact, the negative control, I presume you mean control where there were no cells, just spions. Yeah, results were subtracted from those results to like exclude the effect of spions on absorbance of light itself. So those results are in fact absorbance showed by cells with spions minus absorbance of spions itself. So that's how we prepared our charts.
Okay. Thank you very much, guys. We don't have time, so maybe next time. Thank you. And now we will going to sixth presentation presented by Radosław Dudczak.
Ladies and gentlemen, Mr. Chairman, my name is Radosław Dudczak, and I have a pleasure to present you today with the work entitled Effect of Alpha-Lipoic Acid on the Early Stages of Diabetic Retinopathy, a Pilot Study. So, diabetic retinopathy is traditionally defined as a microangiopathy with hyperglycemia causing damage of blood vessels of eye fundus. And diabetic retinopathy is a very common complication of diabetes, as it is estimated that 1/3 of patients with diabetes will develop diabetic retinopathy. So after blood damage are damaged, traditionally it's recognized that there is VEGF production going, that we have microhemorrhages, microaneurysm with consequent vision losses. But again, this is just a traditional definition, yet still recognized by, for example, ICO or WHO. But since around 60s, we know that in diabetic retinas, in diabetic retinopathies retinas, we have a signs of neurodegeneration processes. And nowadays, we are taking even one step further, a step that may completely change our view on diabetic retinopathy pathophysiology. Because some scientists in recent years proves and theorize that diabetic retinopathy is in fact a neurodegenerative process. So a neurodegeneration is a first step in this disease, and then comes blood vessels damage. Why is that? Because nowadays we know that in retinas, just like in brain, we have something called neurovascular unit. That is combined from elements of blood vessels and neurons. So disruptions in one element causes disruption in second. So with those new discoveries, new questions are raised. So for example, is it really true that neurodegenerative processes are first? And if so, should we still use, for example, fundus imaging as the primary detecting method of diabetic retinopathy? Because even the smallest disruptions in blood vessels of eye fundus may in fact be a sign of advanced disease. So we tried to answer some of these questions, and the aim of the study was to examine basic mechanisms underlying the development of DR and to understand whether alpha-lipoic acid, a natural antioxidant, affects the survival and function of neurons in retina. To do so, we used 30 mice that we divided into two groups. So we have healthy mice and then diabetes mice. Diabetes was induced by streptozotocin diabetes model. Then those diabetes mice were further divided into two groups. So we have just diabetes group and diabetes mice supplemented with alpha-lipoic acid. And finally, Western blot immunohistochemistry were performed. And we should also remember that at every stage of this experiment, also electroretinography was regularly performed. So here you can see the results of electroretinography. In each column, you have two graphs. First graph is for right eye, and the second one is for left eye. So as you can see in the group B, which is diabetes group, we have a very visible decrease in all oscillatory potentials with intermediate protective effect of alpha-lipoic acid in group C. And here, again, you can see the exact average values of each oscillatory potential for each eye. And as you can see, the most visible changes are in oscillatory potential 3. Secondly, immunohistochemistry was performed, and we use appropriate markers. So RBPMs for retinal ganglion cells, beta-3 tubulin, as well as NeuN for neurons, and GFAP for glial cells. And as you can see, we did not observe any structural changes in retina. And finally, Western blots. We were trying to establish various proteins expression, for example, interleukin-6, VCAM, TNF-alpha, but I've chosen the most important ones for you. So starting from the left, we have synaptophysin, and we see a very visible decrease in diabetes group with a protective effect of alpha-lipoic acid. Then we have interleukin-1b expression with increase in diabetes group and with small protective effect of alpha-lipoic acid. And finally, the FAGF expression that is unchanged in diabetes group. And interestingly, there is a very visible protective effect of alpha-lipoic acid. And what does it tell us? That we have currently no neovascularization. So when we link all of these results together, it all becomes quite logic and quite connected. Because firstly, we see that we have in early stages of diabetic retinopathy, that we have functional changes in retina, we have no structural changes, and we have no neovascularization. And what does it tell us? That diabetic retinopathy, in fact, starts as a neurodegenerative process. Moreover, it probably starts with disruptions in synapses. Why? Because, as you can see, we have a very visible decrease in diabetes group in synaptophysin expression. But you may ask, why is that? That here we can see the very visible protective effect of alpha-lipoic acid. And if we would go back to the electroretinography, there was a protective effect, but not as strong as here. Well, we don't know why. It wasn't proved in this experiment. We may only suspect that it's because diabetic retinopathy starts not only in hemical synapses, but also in gap junctions. So this is a very interesting target for next experiment. And just a few words about clinical implications, which are very, very important, because we give you an evidence for that diabetic retinopathy starts as functional alterations in retina. So we shouldn't use fundus imaging. We should use electroretinography or optic coherent tomography, and we would prefer electroretinography as the functional changes are the first. And the second very, very important clinical implication is that if you want to test your patient with electroretinography for diabetic retinopathy, you should double check if he's taking some antioxidants, because if he's taking those antioxidants, they may mark the ongoing diabetic retinopathy process. So thank you all for your attention. If you have any questions, feel free to ask them, and I yield the floor back to the Ms. Chairman.
Okay, thank you very much. Good job. We have some questions.
Yes, I have a question regarding the alpha-lipoic acid and the study in other works. I'm sure that in PubMed you find a lot of information, but what was important for you to choose this alpha-lipoic acid in your study?
Well, because alpha lipoic acid was tested before in diabetes. And in fact, it was the antioxidant that showed us the best effect in diabetes. So we decided to start from alpha lipoic acid in diabetic retinopathy.
Thank you. First, shortly, advantages and disadvantages of the mice model. First question.
Okay, so in our case, we didn't have a choice, really.
Okay.
Because we wanted the transgenic animals to further experiments.
Okay. Second one. I think you should create your conclusions more carefully, because to conclude that there are no structural changes, you should estimate the ECM products of degradation, glycosaminoglycans, proteoglycans, collagens, for the future task, for example. That's all. Thank you.
Thank you.
Hello, thank you very much for your presentation. I would like to ask you about the statistic analysis, because, for example, in the slide where we saw the Western blot analysis, I couldn't see the error bars, for example, and I would like to know how many animals did you analyze and which statistic test did you prepare? Thank you.
Well, starting for the beginning, we have standard deviation here on the graph. We used in total 30 animals. And then for Western blot, we used 5 animals, sorry. Yes, you're right. Sorry. Yes, so we used 30 animals, and for each group we used 5 animals, and the material was combined together because you can get very small amounts of material from one retina, and it's standard. So yes, we didn't perform more advanced statistics because this is just a pilot study and we have a small amount of mouse. So we could do that, but it just, for me, it's art for art at this stage, because it doesn't tell us anything, to be honest, those results.
Okay, we need to end your presentation, but thank you very much for your work.
Yes, but I have a question about the contribution.
So I was taking part in Western blot analysis. I was taking part in immunohistochemistry and in electroretinography. I was also using an imagery software to gather all information for you, or all results, and that's all. But I have to notice that during, for example, Western blot and during immunohistochemistry, I wasn't obtaining retinas because I couldn't. I need more experience to do that, and it's quite hard for me.
Okay, really, thanks. Okay, give a chance another participants. So we have 3 works left today. So 7th presentation presented by Tiago Sousa with online Zoom platform. Do you hear us?
Yes, I'm hearing you. Do you see me?
Yes, you can start.
Yes, can you? Okay. Now I will start. Hello, everyone. My name is Tiago Sousa, and I'm a 4th-year medical student from the Faculty of Medicine of University of Coimbra in Portugal. Greetings from Portugal. And today my presentation is about my investigation at the Center of Neuroscience and Cell Biology at University of Coimbra. So, slide. Okay, slide. Okay, so we have talked about a lot of cancer, which is really important, but I have another disease to talk to you, which is dementia, particularly Alzheimer's disease, an uncommunicable disease, which is particularly important because it's a progressive neurodegenerative disorder marked by cognitive impairment. And we know the physiopathology just a little bit. We know it's incurable, and we also know that we are still struggling to find a cure to this disease. So why this work and why this compound, the 2,4-DNP or the dinitrophenol, if you want to, because 2,4-dinitrophenol targets mitochondrial dysfunction, and we know that mitochondrial dysfunction is involved in the first steps of Alzheimer's physiopathology. And 2,4-DNP, it's a mitochondrial uncoupler used previously to in high doses for weight loss, and now we want to see if he has any important and relevant neuroprotective effects on this pathology. So what we want with this work is to evaluate if the agent is capable of not to revert Alzheimer's disease pathology in vitro using SH-5Y cells. Okay, so let's go. Okay, important, we used an in vitro model. The retinoic acid differentiated SY5Y cells. And how we do that? We culture these cells with a retinoic acid during 8 days until we get differentiated SY5Y cells. You can see on the right side that on the 8th day they were differentiated because they express immunohistochemistry markers for mature neuronal cells. And after that, we exposed to oxalic acid and or 2,4-dinitrophenol for 6 hours. Okay, and we know that oxalic acid acts as serine threonine phosphatase inhibitor, okay, type 2A inhibitor, and we also know that this compound is used to mimic Alzheimer's disease pathology. So we evaluated the cellular viability using the MTT assay, okay, that we all talked about here in our presentation because it's really important. We also try to evaluate the citrate synthase activity because it's a marker for mitochondrial mass, and we also use aconitase activity. Aconitase activity, it's a mitochondrial enzyme sensitive to oxidative stress, and it's really important to have a marker for oxidative damage. We also use Western blot also to evaluate some proteins that I will talk to you later. Okay, and now, and just to close the material methods, I will say that we evaluated MitoPY1. That it's really important as a marker also for oxidative stress and the oxygen consumption ratio and ATP production with our seahorse. Let's go. The results, MTT assay, we show that oxalic acid decreases the cellular viability, and DNP was able to revert this process, which is incredible. In B, the MitoPY1, we can say that oxalic acid really increases, but DNP is also capable of reverting. And this effect was also observed in aconitase activity. When we see that oxalic acid is able to decrease aconitase activity, and DNP was able to revert this effect. The tau hyperphosphorylation in D, you can see that oxalic acid as as a promoter of tau pathology, it was able, in the presence of DNP, it was able to revert this hyperphosphorylation. Slide. Okay. You can see now in E and in F, because we use the same machine, the CRs apparatus, we can say that oxygen consumption ratio was decreased with oxalic acid, and it was reverted in the presence of DNP. And the ATP production, it was also reverted. And the citrate activity, as I said, as mitochondrial mass important enzyme in the cell, and it was also reverted in the presence of DNP, and also the important SDHA enzyme present in oxidative phosphorylation. It was also reverted by DNP. Let's go. That dynamics, as we know, tau AD pathology favors a high increase of efficient proteins, and DNP was also capable of reverting that process and decreases, you can see in phospho-DRP1, decreasing the levels of this protein. Just to conclude, is DNP a promising treatment? I hope so. It needs more studies, it needs more pathways to evaluate if this compound is really capable of doing that, and it needs another models and another projects to evaluate that. And that's where future investigation lives in future projects, exploring the new pathways and really see if this compound is able to really revert this process. Okay, so greetings from Portugal. You can see you can see our location here, and you can see our university and our study group. Thank you very much for this opportunity to be with you.
Thank you, Tiago, for your presentation. Congratulations. And we have some questions for you.
Please describe shortly limitations of your study.
Yes, the study has some limitations. You can see that, okay, but DNP was able to reverse the presence of oxalic acid. But how about any other, I can say, toxins or proteins that are able to mimic Alzheimer's disease? Are they? Is DNP able to revert that? I don't know. Future investigation is needed. I can say another. For example, we incubated DNP and oxalic acid at the same time with the cells.
We got it.
Okay.
Thank you very much. Another questions? Maybe online?
Yes, another question is the same about contribution, because we need the answer.
Okay, thank you, Johanna, right? Okay, okay, Johanna, thank you for your question. Yes, my contribution, I helped designing this project, and I help with the laboratorial experimentation, evaluating these assays. Okay, thank you really much for your question.
Okay, so that's all. Thank you very much again.
Thank you.
Okay, and now please Wiktoria Suche and Katarzyna Chwalebna will present for us the presentation about the energetic nervous system and pain participant in on-activity both anorexia animal model.
Good afternoon, ladies and gentlemen. My name is Wiktoria Suche, and I'm from Jagiellonian University Medical College in Krakow. And today I will have the pleasure to present you our work about the enteric nervous system and pain perception in an activity-based anorexia animal model. To start, I would like to bring you a little bit closer to the main subject of our research, which is anorexia nervosa, AN. It is a devastating eating disorder characterized by extreme food intake restriction. It is often associated with excessive physical activity, which leads to rapid weight loss and, as a result, obviously, to low body weight. People suffering from this condition also have distorted body image, and they are afraid of gaining weight. Anorexia has the highest mortality rate among all the psychiatric illnesses, and interestingly, this mortality seems to be higher among men than women. The vast majority of these deaths can be attributed to a number of complications which affect almost every body organ, and those are metabolic, cardiovascular, neurological, and gastrointestinal symptoms, such as bloating, constipation, or early satiety, which might be the first noticeable physical symptoms of the disease. And now let's move to the experiment. After 7 days of acclimatization period, we divided our rats, 24 rats, into 2 groups, the control group and the activity-based anorexia, ABA group. And then we performed the pain tests, the von Frey test for the mechanical stimuli and hot plate test for thermal stimuli. I can elaborate on them later in the discussion part if you're interested. Then the actual part of the experiment began. And while the control group had ad libitum feeding schedule and no access to the running wheel, the ABA group had the complete opposite. They had the restricted feeding schedule of 1.5 hours a day and the access to running wheel. And the experiment was discontinued after the humane endpoint criteria were met. And then we performed the second series of measurements in the pain tests. And also we euthanized the animals, collected the samples from the jejunum and analyzed them. Now let's take a look at the results. So the differences in body weight change between the groups can be easily attributed to the differences in feeding schedule, which was restricted in the ABA group. However, particularly intriguing is the pattern of increasing physical activity in the ABA group despite the decreasing body weight. And this phenomenon mimics the course of the anorexia in humans very accurately. Next, we have the routine hematoxylin eosin staining, and it shows that in the ABA group, we have the decreased thickness of mucosa and the intestinal wall, as well as worse organization in all of its layers of the intestinal wall in the ABA group. Next, we performed the immunohistochemical analysis, and first we performed it with antibodies against CFOS, which is a marker for neuronal activity, and also against nitric oxide synthase, which is a marker of nitrogen neurons, which are the main inhibitory neurons in the enteric nervous system, and also against choline acetyltransferase, CHAT, which is the marker for cholinergic neurons, the main excitatory neurons in the enteric nervous system. And here, wait a moment. Here we see the decreased neuronal activity in the ABA group. The ABA group is below the control group. Okay. The next staining was against PGP9.5, which is a cytoplasmic protein, and it is just used to generally visualize different populations of neurons, and also against either NOS or CHAT. And here we see, wait a second, it's okay. And here we see the decreased number of neurons of both nitrogen and cholinergic neurons. We can't see. Okay, sorry, but here I can see it a little bit better, that there is a significant difference between the control group and the ABA group in terms of density of the cholinergic and nitrogen neurons. As for the pain perception tests, we observed the decrease in latency time in the von Frey tests, which means that the pain threshold for mechanical stimuli decreased in the ABA group, with no such differences in the control group. And also, in terms of studies on humans, it is a little bit controversial because some of the studies showed that this pain threshold for mechanical stimuli is elevated in anorexia patients, and I can say a little bit more about it in the discussion part because I don't have time right now. And for the hot plate test, the latency time increased, which means that the pain threshold for the thermal stimuli also increased, and this is in accordance with what we can see in studies on humans. And to conclude, apart from the complications of anorexia that affect the central nervous system, we can also observe some complications affecting the peripheral nervous system, namely the enteric nervous system. And alterations such as reduction in the number and the activity of neurons in the enteric nervous system might be the potential pathomechanism of gastrointestinal symptoms, as I mentioned before. And okay, I'm finishing. And we also studied the pain perception in the anorexia, and the results are very interesting because it shows disequilibrium in the process responsible for responding to either thermal or mechanical stimuli. And our work might indicate a direction for future research in this field, and also a chance to develop new therapies for treating anorexia. Thank you very much for your attention.
Okay, thank you very much. And now it's time to start question.
Thank you very much for your presentation. Could you say something more about hot plate test?
Yes, of course. So it comprises like a device. It's called a hot cold plate, and it can be either you have the range of the temperatures that you can preheat the plate to, and it looks like a huge specimen tube, something like that. It is in the shape of a cylinder, and we preheated the floor of this tube to 50 degrees Celsius, and we just put the rats into that tube, and we measured the time between placing the animals in that tube and the moment when they started licking their paws or vocalizing, and it was the sign that this stimuli is nociceptive to them.
Okay, thank you very much. And second question, how many rats did you use?
Excuse me?
How many rats?
24.
Ah, okay. Thank you very much.
Okay, any other questions?
Yes, I had the same question, and now I need the answer. But again, your contribution?
Yes, so I am more into numbers. So I was more of the part in the measuring the body weight, also checking the number of evolutions in the wheel, as well as the pain perception test. And the histopathological analysis was mostly part of my colleagues, Kasia and Mateusz. And also I performed the statistical analysis of the obtained results.
Thanks.
Okay, we have more questions.
I have one question about the pictures you showed to us, so the microphotographies of the small intestines.
From jejunum.
Okay. So you firstly showed us the hematoxylin and eosin stained samples, and then we saw some immunofluorescence. Yes. Don't you think that it would be beneficial to counterstain the cell nuclei or also the cytoplasma to show exactly where the presented markers are situated in cells? Because on the pictures of the fluorescence, we see only some green dots, red dots, but we don't see where are exactly the cells. And also, have you performed some internal control, like isotype control staining, to be sure that the signal you are seeing is the signal from the antibody?
Yes, so to the second question, it was actually something that the reviewers told us about, that we didn't perform it. So the answer is we didn't perform it. And to the first question, yeah, this I think would be beneficial, but I didn't quite catch the first part of the question. It was like to visualize the cells. Oh, the cell nuclei. Yeah, that would definitely be beneficial, but yeah, we didn't do that. We just didn't have the device to show.
Few words. I think that you could see the differences, but I think that now we might not be able to see due to the resolution of the screen. So I think that you could see the differences in your lab or when you saw the results first.
Yeah.
Okay, we need to finish your presentation. Thank you very much.
Thank you.
Okay, and finally, I would like to ask to present the last presentation today's session. Rasyan Salami. I'm sorry if I say it wrong. Do you hear us? Or Władysława Takariewa? Okay. Maybe wait a minute. Okay, so thank you all of you for your presentation. Thank our jury, and once again, we're really sorry for the beginning of this session. And we congratulate all of the presenting authors and participants of the basic science, biotechnology, and biomedical and engineer. At this point, I would like to ask Professor Paweł Olczyk for a few words to sum up the session. Just a few words.
First, I would like to congratulate the organizers for the, as usual, professional level. I would like also to thank and to congratulate all the speakers for your presence, for your results, for your presentations. It was worthy to be with you and to hear you. Thank you very much.
Okay, thank you for those words. And we would like to cordially invite you to decoration and closing ceremony of SIMC 2023 conference, which will be held in A3 Hall on the 19th of May at 4:15 p.m. And I would like to thank you once again and wish you all the best and have a good evening. Thank you.